Okay, looks like we're live. Hi, everyone. Welcome to another seminar. Today, Michael Reiser speaking. So, Michael did his PhD at Caltech with Michael Dickinson, where you must have been, I actually don't know if this is true, you must have been overlapping with both Vivek and Malamurthy at the same time?
Yes, and Rachel Wilson and Glenn Turner.
Wait, wow. I mean, that's kind of like a good chunk of...
It's the Mount Rushmore of fly neurobiology.
And then he moved to Janelia, where he's done a wide range of of work on vision in Drosophila, from fundamental visual encoding to spatial preference, visual spatial preference in Drosophila. So I'm excited to hear what he has to talk about today, and I'll let him take it away.
Okay, great. Thank you so much, Adam. I'm going to go full screen. Can everyone see this? Adam, maybe just let me know, because I can't see the chat window at all.
Yes, we can see this.
Okay, good. Okay, so great. Well, thank you very much to the organizers for this really amazing seminar series. I'm really delighted to have been asked by Adam to talk in his really excellent series on invertebrate neurobiology. It's really a pleasure to present our work. I'd also like to thank my work from home colleagues, my family, for giving me some time and space to make a few new slides. And I'd especially like to thank all of you for joining today. I hope everyone is well and staying safe. And thank you for taking some time out of your day to listen to me talk about our work. So I'll do the talk in two parts. The first is going to be a survey, an overview of how how us and others in the field have been able to figure out how flies are able to detect and see motion. And then there will be a short intermission with some eye candy. And then we'll move on to the second part of the talk, which will be more about our more recent work, all of which is unpublished, about trying to understand it and use the methods we've used to understand the encoding of neurons in the visual system, but apply it to the rest of the brain. And so in my lab, we're interested in vision generally. So we work a little bit on motion, we work on color, we work on feature detection, and we work on pattern learning. But today I'll be talking primarily about motion detection. And really one of the amazing things about motion that we tend not to stop and think about, because it's such a ever-present part of our daily life, is that it's not actually something that our eyes have direct access to, right? So our photoreceptors are essentially just reporting patterns of light hitting the retina, and then within the brain, that has to be synthesized and really constructed into a perceptive motion, right? So the brain has to somehow stitch these little snapshots through time and space to extract perceptive motion. And so we've been studying that in the fruit fly. And as many of you would appreciate, one works in the fruit fly because of all of the amazing computational tools, but much less appreciated is the fact that the fruit fly really has exquisite behaviors, right? So we come to this animal because of genetics, but we should not forget that they're actually quite good at a whole range of things. And motion vision is really, I think, one of the places where this organism shines. And so here I'm going to show you a high-speed video from Michael Dickinson's lab of an escape behavior in a freely flying fruit fly. I hope everyone can see this. This has been slowed down by a factor of 300. Right now, just off screen, there's a looming visual object that approaches the animal. And within 60 milliseconds, the animal sees this object, understands, to the extent a fly understands anything, that it is approaching it, and computes an evasive banked turn and flies away, right? So that is a thing which I think all of us have personal experience with, but slowed down like this, you appreciate really what an amazing feat of, let's call it neural engineering, goes on in the tiny fly. All right, so this is exactly the kind of behaviors we wish to understand from a computational level, and then later with circuits. And so that's what I'll talk you through some of this today. And so if you think about the challenge for the animal and subsequently the challenge for neurons in the animal's brain, it is really to extract the fact that this object is approaching it, right, looming towards it. And you can imagine there are multiple ways to extract that looming information, but a particularly reliable one would take advantage of the ability to extract the direction of motion of the edges, right? So you have this radiating outward pattern, and so if you could pick that up computationally, you certainly would be able to estimate both the location and something about the other features of the looming system. And so what we've known for a long time in insects is that the motion computation is local. And so what we mean by that, and this was arrived at through behavioral experiments in the 50s, pioneered by Haschenstein and Reichardt, is that the motion detection itself is carried out in a very small neighborhood on the eye. And so for fruit fly, that is something on the order of about 15 degrees, a little disc with a diameter of about 15 degrees. And so that's quite convenient because it means that as we start to hunt through the animal's brain for the neurons that carry out the computation, we already have a very specific anatomical and functional footprint, right? So we're looking for neurons that image about that much of the world. And so the essence of the computation turns out to be even simpler. So for illustration, let's just compare this minimal case where all we're doing is thinking about the input striking two facets of the fly eye. In this case, I've just enumerated them as location 1 and location 2 here. And so in the case of this looming edge, you can imagine that if you have this sweeping disk radiating across the eye, right, that you might want a computational element, right, which will later will be a neuron in a circuit, that is able to encode the direction of motion in what by convention we'll call the preferred direction. And so you would like to contrast that with receding motion, which is moving in the opposite direction, which by convention we'll call the null direction. And so really the goal for the field is to report directional selectivity, which is the term of art for this computation. And it really boils down to something as simple as being able to report that location 1 has seen the luminance change before location 2, as opposed to 2 saw the change before 1. So that would disambiguate between the expanding and the receding looming stimulus. Right, so there are really two general strategies for how to report the direction of motion. One is that you would enhance the response for the preferred direction, and the other one is that you would suppress response in the null direction relative to the opposite direction. And so these are the two strategies, and they map onto two classic models which have inspired, these are theoretical models that essentially imagine what the computational circuit might look like. And so these map on to these two models. The first is the so-called Haschenstein-Reichardt detector, which is the canonical example of a preferred direction enhancing element. And then the canonical example of the suppressive one is the so-called Barlow-Levick detector. And so rather than talk you through all the details here, I'm just going to animate them. I hope everyone can see this, just to give you an idea of how they work. And so here, if you imagine a little spot of light passing over location 1 in our schematized retina, it's going to pass through this delta t, a time delay. And so that is a slower arm of this detector. And then at location 2, it passes through a non-delayed arm. And if the timing works out, you end up with this coincident arrival of these excitatory inputs. That's what's indicated by the 2+ signs. And then they have this synergistic interaction indicated by the multiplier. So you get a positive, strong response. Now, when motion moves in the opposite direction, the null direction for this detector, you go through the fast arm first and then the slow arm. And so, of course, you no longer have this coincident arrival. So there's no overlap, and so a minimal response. So that's the logic behind the Reichardt detector. Now, the Barlow-Levick detector works differently. It combines a fast excitatory input with a slow inhibitory input, which is offset. And so if you see if motion is moving in the preferred direction for this unit, you have this, essentially the excitatory input sort of slides through that rectifier at the bottom. And when motion is moving in the opposite direction, you initially kick off this inhibition, and the inhibition is powerful and long-lasting, such that it can cancel out the subsequent arrival of the excitatory input. Right, so that's the logic of the Barlow-Levick detector. Okay, so with that framework in mind, the goal for many people was then to look at circuits in the fly brain and to identify the neurons that are actually computing the direction of motion, and then to evaluate just to what extent these theoretical considerations actually explain the operation of those units. Now, in the fly visual system, we're really aided by having access, and I'm going to try this pointer. I hope it works. So we're aided by, hopefully you can see this, that the structure of the fly visual system has these really prominent retinatopic units, columnar structure of the visual system. We'll get to retinotopy in a second. And if I just bring on some labels here, you can see that we have in the middle here the central brain, and off to the side is what in insects we call the retina. So that's really just the part of the eye that doing, let's say, the business end of light collection, right? So all these light guides down here. And then we have this prominent chiasm, but the really nice experimental feature of this system is that it's composed of repeating retinotopic units. And what we mean by that is that if you imagine a part of the retina that is imaging a small patch of space, the subsequent stages of processing that patch of the visual scene will be happening in neighboring elements throughout all of these layers of the visual system. And in flies, the visual system is usually referred to as the optic lobe, and it's comprised of all of these different neuropils. Okay, so that is sort of a anatomical view. And so now switching to a schematized view, this is really, I'm going to just walk you through what the field has learned over the last 15 years. So about 13 years ago, when I started my lab at Genelia, we knew next to nothing about the internal logic of the motion pathway. So here in the schematic view, I'm just going to summarize the key transformations that happen within a single column of the motion pathway, but bear in mind that it's actually repeated throughout the eye, right? In these columns, which correspond to each facet of the compound eye. So from classical work, let's say from the 60s until this century, right? We understood, and this was largely done in larger insects, mostly in blowflies. We understood something about the two ends of the visual system, right? So we understood that at the output level, down at the bottom here at the lobular plate, we have these large field motion sensitive neurons, which appear to receive inputs from directionally selective neurons. And then what was really well characterized is that there were these transformations from the photoreceptors in the retina that project to the feed-forward neurons of the lamina that signal changes in light intensity to the rest of the visual system. Now, if you saw Marion Seeley's excellent talk on Monday, you'll know that this view is a little simplistic, and there's more to be said about what's going on in the early stages. But so our focus and what most of the labs who embraced these questions in Drosophila starting about 15 years ago, and I've just listed a large number. I hope I've gotten everyone, but there are really a large number of contributions from many really excellent, wonderful, clever, creative scientists who've helped fill in this picture. And I should just say, as a field, we've had occasional disagreements, but by and large, I would say there is a much more consensus than anything to argue about. And so one big, really key piece of the puzzle that filled in first was this idea of on-off selectivity, which had previously been mysterious, but it turns out that also in the fly visual system, we have this strong bifurcation into on and off pathways. And so just so we're on the same page and everyone knows what we're talking about, if you imagine, let's just say there's a hypothetical experiment we're doing with a visual stimulus. So if we just show, if we have a gray background and within a patch of that, we have a luminance increase, which is what we nominally call on, neurons within the on pathway would be driven by that sort of stimulus. And conversely, in the off pathway, we just have this local darkening, right? So columnar neurons along the off pathway would preferentially be driven by that darkening. And so there's an internal feature of the visual system, which is why it was unclear before, right? If you are working in the retina or in the lobular plate, you wouldn't actually be aware of this on and off segregation. And then the other big piece of the puzzle was figuring out and identifying that directional selectivity emerges first in the so-called T4 and T5 neurons. So this is down here at the bottom, and it happens twice. So T4 is the on-directionally selective neuron, and T5 is the off-directionally selective neuron. And so there are a very large number of studies and papers, really beautiful work that went into this. I'm just going to show you a very small piece of our own work for how this sort of argument was constructed. And so again, it comes down to comparing the responses of neurons to different visual stimuli. So in this case, now we're going to compare, starting from dark patch on a gray background. We're going to compare an on flash, so just this luminance increment, to an on stimulus generated through a moving edge, right? So the starting point and the ending point is identical, but it's the transition between them which has a completely different structure. And so if you express a genetically encoded calcium indicator in single cell types in the fly visual system and show the fly these visual stimuli and measure calcium responses, what you will see is that in the case of the T4 neuron, the on directionally selective neuron, you get this very strong enhancement for the motion stimulus as compared to the flash stimulus. Whereas if you image from any of the inputs to T4, which is what we did in this 2017 paper, and in this case, I'm just showing MI1 as a representative, you do not get this enhancement from motion. So that is to say all the inputs to T4 are motion blind, whereas T4 is doing something to synthesize motion from its inputs. And so that really was the main question that a large number of labs have tried to pursue. So much of the insights and really the reason we were able to fill in that diagram, a lot of that actually came from connectomics. And so I was fortunate to be working at Genelia, where a number of colleagues focused on the flight visual system, primarily because of its columnar structure, right? So that meant that a small volume reconstruction would have a lot of payoff, right? So it would describe a lot of the principal circuits of the visual system. And for us, it had this particular advantage that by doing entire columns, in this case, 7 columns of the medulla, it contains the entire circuitry of the on pathway, which is why in our lab, we focused quite strongly on T4, the on directionally selective neuron. And so what is required to interpret these medulla connectomes is you have to go through and identify individual neurons, which could be done just based on morphology from classic Golgi studies, and also now with these genetic labeling methods. And so you could work through this data set and identify that the primary inputs to T4 neurons, so T4 neurons are these small little cells here. They have their, I don't know if you can see my cursor. Let's try this again. All right, so T4 neurons are here. They have these small dendrites down here in layer M10, the very bottom of the medulla down here. And they receive inputs from these columnar neurons, right? So these are neurons which are essentially just relaying the signals, in this case from the on pathway into T4. And really what was really beautiful and striking and absolutely convinced me we had to work on this system was this result from our colleagues in the FlyEM team, which is if you look at the dendrites of T4 neurons, what you find is that they receive very patterned inputs. And at each location on the dendrites, they're primarily receiving inputs from a specific cell type. So MI9 is out on the distal tips, MI4 is here on the base of the dendrite, and these MI1 and TM3s are in the middle. Actually, the details don't matter, but what was really exciting about this is it meant that we could use our genetic tools to target individual neurons and then try to probe the functional consequence within this anatomical receptive field of the neuron. So one more important feature I should mention is that T4 neurons are clearly the neurons that support that original behavioral result that motion vision is mediated through comparisons within something like corresponding to about 15 degrees of visual angle. And then one more point is that T4 dendrites are oriented. And so actually this orientation of the dendrites actually reveals the direction of motion that this particular T4 neuron would be sensitive to. And so we'll come back to that in a little bit. And so in this 2017 paper, we went on to carry out a really elaborate, stepwise, complicated study, and it's a bear of a paper to read, and I apologize for that. But what we tried to do was really throw everything we could at this circuit to try to infer the structure of the receptive field anatomically, and then look at the consequence on that for the functional sort of algorithmic reasoning that I introduced before. And so that required making precise genetic driver lines, which we do with our collaborator, Alyosha Nurin, who's quite good at generating multiple nearly perfect driver lines for each of the principal cell types, which provide inputs to T4. And so I'm not going to go through any of those results, but I'll just summarize what we've learned. And so what we've learned, again, inferred from the circuit analysis, which has recently been backed up with gene expression data. And so this all looks very solid, is that within the receptive field of a single T4 neuron, the primary inputs in the center are excitatory. And so that means that for thinking about something like a Hutchinson-Reichert detector, one could imagine that you could have a mechanism, some combination of a dendritic, an intracellular process, right, a synaptic mechanisms that would facilitate this enhanced response to the preferred direction of motion. However, the circuit analysis also shows that towards the base of the receptive field, one finds inhibitory inputs. And so that also means that if we're thinking about the Barlow-Levick type mechanism, we could well have the interaction between the two sides of the receptive field, which could facilitate a suppressive mechanism. Right, so both of these seem possible. So we did all of this work on T4, and at the end of this analysis, it wasn't actually clear. And so what seemed most likely to us at the time was, in fact, we had some sort of hybrid detector. And so we proposed that in the 2017 paper, and it was quite popular at the time. There were, I think, nearly every lab in the field claimed in their paper that what looked most likely was that, in fact, biology took advantage of both solutions, which I think is a narrative that people are somewhat comfortable with. And so it all made sense, right? If one is a good idea, if both are good ideas, do both of them. However, we couldn't actually be sure. And there were some inconsistencies in the data that didn't totally check out. And so I would say the main reason that we weren't sure is because the way all of the labs in the field were doing this analysis, was using calcium imaging. And calcium imaging is a great method, but there are some issues. In particular, for this kind of discrepancy between the models that we're trying to make here, one would like to know, for example, are the inputs delayed at different locations on the dendrites? And the scale of time delays we care about are well under 100 milliseconds, right? So you can't resolve that with a calcium indicator. Furthermore, we also really want to confidently ascribe a functional role to inhibition. And again, calcium indicators are quite insensitive to hyperpolarization, especially in the dendrites of these neurons, which at rest have very low calcium levels. So it's nearly impossible to detect reductions in calcium. And so Eyal Gruntman, a really brave, heroic, talented postdoc in the lab, took on the challenge of trying to get reliable whole cell recordings from T4 neurons. And so I should say these are among the smallest neurons in the fly brain. They're also the most numerous neurons in the fly brain in terms of cell types. And so they're clearly important. And we were excited to try to figure out what they do. And so not only did this require reliably getting recordings, but then we also had to reconceive of our approach to generating and delivering visual stimuli. So to do something much more like single unit neurophysiology done in primates, right? And so this required online mapping, identifying receptive field centers, then finding the axis for which the neuron is most sensitive to motion, and then mapping within this reduced one-dimensional receptive field. So all of this work is published in a paper from about 2 years ago. So I'm just going to walk through a single key result from the paper. And so with this framework of thinking about the anatomical receptive field. And so if one delivers, remember, this is an on-directionally selective neuron. So if one delivers bright flashes along this one-dimensional receptive field, you'll find depolarizing responses from these neurons. Now I should mention these are non-spiking cells, so we get a depolarizing response. And then if we sort of march along the receptive field towards the center, we get an even larger depolarizing response, which is practically, that's how we define the center of the receptive field. And if you move towards the trailing side of the receptive field, you find this compound response, which is excitatory at first and then inhibitory. And then you'll notice even at this level that we have fast excitation and slow inhibition, which gives us a clue about what might be going on. But in order to really test this, what one needs to do is to take advantage of a fairly standard approach where you then start to combine the pulses delivered at multiple locations with doing so-called apparent motion. So if you first deliver a flash on the leading side and then in the middle, you'll measure this compound response, which looks approximately like just the linear sum of these two things shifted in time. And then if you deliver the same stimulus in the opposite direction, middle, then leading, what you'll find is, again, something that looks roughly like the linear sum of those two responses, roughly what you expect. But importantly, if you compare the amplitude of the large responses here, you'll see that they're exactly the same, right? And so what that means is that if stimulation is restricted to the excitatory lobe of the receptive field, this neuron is completely motion blind, right? Now, we're going to contrast that to the same kind of experiment done going from the middle to the trailing, and then again now in the opposite direction. And here is where you finally find this large suppressive event, right? So the first time we actually see any asymmetry is only when we combine stimulation between the excitatory and the inhibitory part of the receptive field. And so what we find is that we have this very strong suppression of motion in the null direction. And then through a whole bunch of analysis we've subsequently done and in a follow-up paper, we are pretty sure that this is the primary mechanism for motion detection. There's very little data that this can't explain. And so I'm just going to give you one flavor of the kind of follow-up analysis we've done. And this is to take a data-driven computational approach with our wonderful, excellent, brilliant colleague and collaborator, Sandro Romani, who's a computational neuroscientist. And so Sandro helped us conceive of a framework and then implement a conductance-based biophysical model of a T4 neuron. We actually use a real morphological reconstruction of a T4 neuron from EM, implemented this in Neuron, and used our own data to fit this model. And so the general exercise is very straightforward, right? It's a question of generalization of the model. And so what we do is we take the model and use it to fit what we call the flash responses, right? The kind of data I previously showed you. So how the neuron responds to flashes at single positions along the receptive field. So I only showed you 3. We do a lot more than that. We densely sample the receptive field. And so you can see that the model can fit this quite well. So that's good. But now as a test of generalization, we ask whether the model can actually predict responses to stimuli that never went into the model. And the relevant one here is motion itself, right? So we only gave the model, these static flashes. And now we asked, can it integrate the inputs, the conductance changes from the static flashes into a directional signal? And so, of course, we did the experiment as well. And so when we measure that, we find what we expect, which is a large directionally selective response, much larger depolarization for preferred direction than the null direction, here at a particular speed, about 30 degrees per second. And we simulate that with the model, and we find essentially nearly the identical response, not just in amplitude, but also in the dynamics. And that holds up across a range of stimulus speeds, right? So, so that really gives us confidence that the mechanism we seem to have our hands on here that is captured in this model is explaining the bulk of the directional selectivity one sees in T4. And so just to summarize this piece of the talk, what we see is that we have a mechanism which is surprisingly simple. It's really just fast excitation within one receptive field and then an overlapping receptive field of slow inhibition. So just putting this in the format of the classical models as a two-arm model, it would look like this, these overlapping receptive fields, and then a fast and a slow arm. And then we need a biophysical neuronal integration model. You can't simply add up the pluses and minuses, right? You actually need to integrate the conductances. And so the logic is very much in line with the model of Barlow-Levick, but the implementation and the details are different in a few interesting ways. And so, of course, I think what you'll notice is that this looks quite good to us because it really sort of matches what we understood about the receptive field from the circuit analysis. And so I'll come back to that in a minute. The other thing I was going to say, and this is something we've started to appreciate more and more in the last few years, is that this mechanism actually looks better in many ways than the classic one. So the classic Reiser detector, which was derived from insect work, turns out to be not particularly efficient. So I'm happy to talk about this later, but generally what I mean by this is if you just simulate our T4 model with the bar moving through it, as I showed you, it'll be much more selective for the preferred direction. And if you take a Reiser detector and implement it with parameters that are matched to what we find in T4 neurons, so if it has to operate under the constraints of the T4 neuron, what you find it is also directionally selective, but is much less efficient. So it is less selective, and it would essentially have to waste all of the signaling capacity generating a nearly largely similar response to null direction motion, which would then have to be cleaned up by downstream comparison circuits. And so not only do we find something which is simple, it actually turns out to be elegant and efficient in ways I wouldn't have predicted before we started. Okay, so we felt quite good about the T4 version, and so we presented this work at some conferences. There's a little bit of controversy in the field, which if people want to ask about it, we can talk about it at the end. So we absorbed all of this feedback and we tried to respond to it, not by going back to T4, but we thought, let's record from T5. So T5 is the off directionally selective neuron. And so we have a recent paper that came out in Eli from Eyal's work in T5, where we took really the same approach, but I would say we were buoyed by all of the feedback. And so we thought, continued thinking hard about stimulus design and expanded the approach and the modeling. And so we really had no strong expectation initially. I think whether we found T5 to do exactly the same thing as T4 or whether we found it to do something different, both would have been interesting. But to cut to the chase, what we find is T5, the off directionally selective neuron, as long as you flip the stimuli from on bars to off bars, you essentially find the exact same structure of the receptive field with depolarizing inputs on the leading side and these compound, including hyperpolarizing inputs on the trailing side. And so really the T4 model, again, just applied to T5 and with the luminance polarity flipped, now predicts a wide range of stimulus responses all the way up to gradients. And so this is all quite good and suggests that really we're on the track to understanding this mechanism, except where this all starts to fall apart a little bit is that as we understand from the circuit analysis and from gene expression data, all of the small field T5 inputs, which I haven't talked about, right? But T5 also receives collinear inputs, and all of them are cholinergic. And so in the fly, that generally means that they're excitatory. And so what we find is we find two neurons, T4 and T5, that implement the exact same mechanism, but apparently are doing it with different inputs. And so we have a long and interesting treatment of this in the recently published paper. So I'm just going to just lay out, there's a little bit of ambiguity here. I don't actually know what the right answer is, but I just wanted to mention how we're thinking about this now. So there are a few possibilities. One is just that maybe it's not direct inhibition, in T5. Instead, what if you have some tonic inhibition that the neuron gets, and in a stimulus independent way? And as the stimulus sweeps through, what we get is we're getting actually the suppression of an offset excitatory input, so something that happens upstream. So that is possible. And so we've handled that with modeling, but it seems unlikely. And so we have an interesting treatment of that in the paper. The other possibility, which is a really exciting and interesting one, is that the fly actually has another input to the directionally selective neurons, which I didn't tell you about, which is one of the craziest neurons I think anyone has ever seen. This was discovered, described, and named by Alyosha Nern, our collaborator. He's called it CT1 for complex tangential 1. And so this is actually just a single neuron. The fly has two, one per side. And the cell body sits right here in the central brain in between the two antennal lobes. And you see there's this giant axon. And then it does this crazy thing. It splits into two fields, one which innervates the lobula, one that innervates the medulla. And this neuron has terminals which contact every single T4 and every single T5 in the fly visual system, right? And it is a prominent input, and it is also GABAergic. And so initially we thought this was a wide field neuron and was unlikely to have this role of delivering stimulus locked small field inhibition. However, Axel Borst's lab recently showed that in fact, if you image from the terminals of these neurons, they do act like small field neurons. So this very much remains possible. All I can tell you is that in our lab, we have not been able to genetically silence this neuron and find massive deficits in visual motion. But there's really, clearly we should understand what this neuron does. And then the last possibility I was just going to mention and hopefully show some pretty pictures before we take our break is that some of the cholinergic inputs could activate an inhibitory conductance. So this would be an exotic, as far as I know, undescribed mechanism. But you know, the fly genome and actually these T4 and T5 neurons contain many acetylcholine receptors that have not really been well characterized. And so we do not have an answer to that either, but I just wanted to mention a pilot project we've been doing in the lab using existing tools was to just try to localize RDL, which is one of the GABA-A receptors, a chloride channel in flies, which is known to mediate fast hyperpolarization. And so we've tried to localize that on the dendrites of T5. And so you quickly run into a problem here. This was a project of Ed Rogers in the lab, which is the neurons are very small, and with confocal, it's quite hard to localize these things. And then there's a general question about how localized the expression is to begin with. So we pursued this localization of expression with expansion microscopy. Ed collaborated very closely with Paul Tilburg, one of the inventors of the method. And so for people who don't know, I recommend you to look at the paper. It's really crazy, but you literally just expand the samples after you've labeled them. And then the labels expand. And so working through a whole bunch of labeling strategies and genetic methods, Ed was able, in this case, to show quite well localized little puncta of RDL within this sheet. So this is now a sheet of T5 dendrites. These are not single neurons. And of course, this has been expanded by many times. And I'm just showing here an example of approximately the same scale of the unexpanded sample. And so I told you it's localized, but it's hard to interpret, and it gets much easier to interpret when we co-label the CT1 neuron, right? So CT1 here has been co-labeled, and you'll see that there's this almost perfect co-localization of the GABA receptor opposing the terminals of CT1, this giant GABAergic neuron. Okay, so this is just to say if you take it away, again, you'll see the RDL right where the CT1 receptor is. And what's really nice about this is we actually are not finding RDL elsewhere, right? So within the T5 dendrites, we're not finding this GABA receptor at other locations. So that again confirms the initial problem we're trying to resolve now, which is where does the inhibition come from? And so CT1 certainly does look like a very good candidate. Okay, so I think, let me just summarize by saying, I would say this is really the remaining challenge for the field, is that we would like to match the circuit mechanism to our functional understanding. And the irony is not lost on me that in the modern era, using all of the Drosophila tools, right, we did an awful lot of experiments where we got, really made a lot of progress, but could not nail down a mechanism. And using a kind of classic method, right, so single neuron neurophysiology and clever stimulus design, we have a lot of clarity. And so the challenge for us and for others in the field is to sort of bring these two ways of thinking about the circuit together. And then I think we can feel quite good about what we've accomplished. And so we're going to take a 3-minute break. Oh no, I'm wrong. I was going to show one more thing. Sorry, I apologize about that. We'll take the break in a second. Okay, so the payoff for understanding how directional selectivity works is that then we can make really strong inferences about what goes on downstream. And so I didn't mention this yet, but within the visual system, right, as a consequence of retinotopy, we have this spatial organization in this dimension. And in the lobular plate, we actually have a map of directions in the other direction. And so again, space is represented this way. And across the layers of the lobular plate, each layer receives the axons of one type of T4 and T5 neuron. And so we now understand the anatomical basis for that from this EM reconstruction. So there you can see spinning around the dendrites of the individual T4 neurons, each pointing in different directions, which is the basis for their distinct directional selectivity. And then the axon terminals are then projecting to these different layers, giving rise to the layer pattern seen in this beautiful imaging result from Axel Borst. And so then if we reflect back on the looming detection problem that I started with at the very beginning, we can then ask, right, actually there's a quite straightforward strategy for doing this, which is if you can imagine there are neurons which are able to integrate inputs in all 4 layers of the lobular plate with just the right pattern, this sort of cross shape, then at least for this location on the eye, you'd have an exquisitely sensitive looming detector. And in fact, that's what we found. So again, Alyosha Nurin, our anatomical hero, colleague and collaborator, identified the so-called lobular plate, lobular columnar type 2, LPLC2 neurons, which have this cross-shaped arborization in different layers of the lobular plate, which we've color-coded in here. And you can see in the spinning one. So there are dendritic fields which radiate outward from a single location on the eye. Now that is a single neuron, and these neurons form a population of about 100 of these cells, which tile the entire eye and project directly to the giant fiber. Right now, the giant fiber mediates fast escape takeoff behaviors in the fly. And so we got together with Gwyneth Card, with whom I share postdoc Nathan Klopotki, who worked on this project to identify how LPLC2 neurons encode looming. And along the way, we found that if you activate these neurons by using optogenetics, the flies jump, right? So as expected, these neurons play a key role in mediating escape behaviors, presumably by transducing these looming visual stimuli. So when Nathan does 2-photon calcium imaging from these neurons when expressing a genetically encoded calcium indicator, he finds, not unsurprisingly, that they respond to looming motion. I would say what was quite a bit surprising was the fact that they were about the most selective stimulus selective neurons we've ever seen. So especially when you compare them to the properties of the inputs, right? If they are really exquisitely tuned for looming to the exclusion of everything else. And so if you're interested in that, I'll point you to the 2017 paper where Nathan did a whole beautiful series of experiments to understand why that is. But what I would say is this is really the kind of thing we wish to understand, right? So taking properties of these neurons within the visual system and then using them to work our way downstream. And this is really, I think, one of the first examples, certainly in the fly, of having an entire pathway, right? Where we go from the photoreceptors to the jump muscles of the legs, where we really understand, I think, the primary transformations at each step along the way. And so this is exactly the kind of thing we hope to do for, ideally for slightly more complex behaviors than takeoff. Okay, so I apologize for that. As I said, I promised you an intermission. So here goes, it's 3 minutes. And I will give you this very nice, this is the expansion microscopy movie. If anyone wants to look at it, I'm gonna go and get a cup of coffee and I'll be right back. Oops. I think I stopped my timer, but we're about, I think 3 minutes is about up. Okay, so I think we'll get started again. Let me go full screen. Actually, let me just skip this slide. Okay, so, right, well, welcome back. So what I told you about before was really just trying to walk you through how we've learned how single neurons detect motion, and then at least one quick example for how the fly brain actually uses that motion signal. And so if we reflect back on, I would say, what the strategy and what actually worked here, well, first of all, it's quite helpful to have a really collegial community of multiple labs who share methods and discuss progress, and a common set of tools, right? These genetic reagents we use for neurons, and connectomics is really critical. But just practically, I think what was really key was being able to localize specific transformations. And so this is something that the field did multiple times. So we identify where on and off emerges, where directional selectivity emerges, and in the case of the looming sensitive neurons, how motion patterns are selected for. And really that discovery, right, this localization, knowing which neurons to work on, really sort of organizes the experimental project to get to mechanistic insights. And so that is, I don't see any reason why that approach shouldn't work for deeper circuits in the fly brain, and that's exactly what we're trying to do now. So again, we're now trying to use this approach to try to understand sensory motor pathways for visual motion guided behaviors. And so what kind of behaviors are we going after? Well, the one that is my personal passion is the so-called optomotor response. So seen here in a walking fly, where if you see on our LED displays, we have these drifting gratings, and we're zoomed in on the fly. I hope everyone can see this. And when the bars don't move, the fly is stationary or walking forward. And then in this case, you'll see the bars will start to move from right to left, and the fly will turn, the ball is moving, and this is the fly turning fixed in space, but turning with the intention to follow the bars, right? So this is a body and gaze stabilizing reflex where the external world is applying this strong visual motion signal, and the animal is compensating for that. And so that's something that we've worked on for quite a bit in the lab. And really what's shocking, especially given that these optomotor behaviors have been so critical to thinking about motion vision in insects and in many organisms, is that the pathway in insects where these studies really began is still unknown. So once you leave T4, T5 land, we don't actually know which neurons mediate this optomotor pathway. So this has been something that we are trying to do in the lab. I will tell you we don't know the answer, but we've learned a couple of cool things along the way. And so we've taken two strategies to approach this. One, I'm going to call follow the behavior. The second is called follow the anatomy. So follow the behavior. What do I mean by this? On flies, we have the ability to design relatively efficient experiments and use them as a screen. And so here's a project we did at Janelia with a lot of help during a project called the Fly Olympiad, which we started just when Janelia opened. And with help, I built this contraption, which we call the Fly Vision Box, in which flies are in these corridors, and we just show them visual motion along the walls. I've slowed it down here, so hopefully everyone can see this. The flies powerfully follow the direction of the visual motion. And so obviously it's quite efficient because you just put the flies in a box, count them out. And I was excited to see that within this apparatus, we could generate and measure behavioral responses that look quite good. And so what I mean by that is we can measure, in this case, just the following behavior of the flies. And it is speed tuned in exactly the same way as one would measure doing single fly experiments with a tethered fly. And so that really gave us confidence to proceed and do a big screen. And so we screened a few thousand of the so-called Gen 1 genetic driver lines where we target different neurons in different combinations. And the problem was actually that we were too successful in some sense. So something like 1 in 5 driver lines would show deficits in these behaviors. And of course, many of them are just silly, like the flies aren't walking particularly well. But practically, it's a challenge to sort this out. And so we spent quite a bit of time doing that with a lot of help from Kristen Branson, who came up with a really brilliant analytical approach to this kind of thing. And so I'm going to ignore all of that and just tell you that the most interesting cell we pulled out of the screen, there are about a dozen, but our favorite one is this projection neuron, which again, Alyosha helped us identify and develop a driver line for. That's the so-called LPC1, the lobular plate columnar type 1 cell. So this is a projection neuron from the lobular plate, the center of motion, into the central brain. And if we look now, instead of at the whole population of LPC1s, but using a multicolor random genetic labeling approach, also developed by Alyosha, you can see that what we have here are small field neurons. That's why we call them columbric cells. So they just cover a small patch of this retinotopic visual space in the lobular plate, and they all project to a glomerulus in the central brain. Okay, so then we take these neurons and do the kind of follow-up experiments we do in the lab. So we silence the neurons, and when we do that, and then take the flies and put them on a ball and do these kind of optomotor behaviors, we were shocked that we find no deficits relative to control. So here, putting a fly on the ball, we look at either the reduction in forward walking when the animal sees a turning visual scene, or the turning behavior, and we really see no difference when we silence all the LPC1s. And so the experience for the flies in these corridors has a lot more translational motion vision than what has typically been studied for flies. And if you just think about your experience walking through a corridor, you do experience plenty of translational motion vision, which has a very different structure than the optomotor stimulus. And so we take the flies on the ball, and then we show them translational stimuli, so front to back or back to front. And what we find is a really striking deficit in the forward walking of the flies. And the flies are completely unable to slow down as a control fly would when they see back to front visual motion, right? So that is the deficit. And so what we find is we have these small field neurons that are required for these reactions to translational motion, but not rotation, which is really surprising. So much of the work in the field has focused on the neurons that are sensitive to rotational motion. And so this is quite different. In terms of the directional preference of the neurons, it is consistent with what we had expected from anatomy. So these neurons have a prominent dendrite in the second layer of the lobular plate, which is the layer which is sensitive to back to front motion. So that fits. Then we do a quick optogenetic experiment. It wasn't quick, but it's straightforward conceptually, except we added a wrinkle where we tried to separate the effects of activating these neurons unilaterally and bilaterally, because we were really keen to make sure to see whether the neurons actually contribute to any turning behavior or really just control of locomotion in the forward and backward direction. And what we find when we do that is when you, in this case we use crimson, but when you depolarize these LPC1 neurons, we get a really striking effect on forward walking. So when you depolarize the neurons either bilaterally or unilaterally, the flies slow down, but has no effect on turning. And because we do this unilaterally, Jessica and Ed Rogers in the lab painstakingly attended to whether each fly had expression on the right or left side. And we can use that to see that actually, even when you account for that, it's not just a feature of averaging, there really is no turning bias. Right? So these neurons contribute to slowing down. So they have this powerful effect on a motor behavior, which for us is quite exciting. As I mentioned, we have no connections between optic lobe output neurons and behaviors outside of the takeoff one I showed you before. Okay, so then we did some imaging from these neurons. So this was the work of Matt Isaacson in the lab. And as expected, if you do calcium imaging and just present ipsilateral stimuli, so record from the left side and show stimuli on the left side, as expected, these neurons are only selective to back to front motion, not to motion in any of the other directions. Again, consistent with the anatomy of the cell. But then it got more interesting. So then if you present full field stimuli, which combine motion on the other side of the eye, what we found is that the neurons are exquisitely sensitive to regressive optic flow, right? And not at all responding to rotational visual motion. So we have this selectivity for translation over rotation, which is really surprising because these are small field visual projection neurons. And so it's unclear how they could resolve global pattern of motion. And so if you just think about the structure of optic flow, which I haven't talked about, but optic flow is really this pattern of visual motion that you could imagine building up from all of these small field local motion detectors like T4 and T5. So if you think about the structure of that motion field for an animal that is turning, like the one at the top here, or for an animal that is translating, like the one at the bottom, you have this very different global structure. But if you're only looking at one side of the visual of the eye, if you're only looking at one eye, you'll see that there are cases where the visual motion is identical, and the confound is resolved by comparing to the other eye, right? And that's exactly what the LPC1s appear to be doing. And so if you, and here I'm just going to build it up. So when Matt stimulates with ipsilateral back to front motion, we get a response, and it's speed tuned in this way. And if you then on the contralateral eye add more back to front motion, you get this enhanced response at all speeds. And if you just flip it so that you now have front to back motion, so this would be consistent with global rotation, we get this strong suppression, right? And again, that holds up at all speeds. And so what it means is that these small field neurons are suppressed by contralateral motion. And so that means that they actually know what's going on the other side of the brain. So that's cool, and really goes a long way towards strengthening the case that these neurons are specialized for translation, not rotation. And so I'm really hoping to get this work out on bioRxiv very soon. It is nearly done. It's been a lot of work and it's taken a long time. But today I just want to tease you with one more thing, which is to point out that these neurons respond happily, vigorously at 9 Hertz, which is really surprising because if we just pull out a single result, so we have similar data as well, but if we just look at the speed tuning of the T4 neurons, here at 10 Hertz temporal frequency, they do not respond to stimuli at all, right? And so somehow it means that these neurons are actually faster than T4 and T5, the directionally selective neurons. So how do they see motion that's faster than their inputs do? And this is not just a curiosity, this is really a critical question and one of the big gaps in how we think about motion and visual motion driven behaviors in flies, right? Is that the behavioral speed tuning, as I showed you at the beginning, is really peaking at 10 or 20 Hertz, right? So this confound is actually really important and needs to be resolved. And so this is an opportunity to just briefly mention the beautiful data enabled by David Bach's lab, the so-called FASB, the adult fly brain EM volume. And so within this volume, we can reconstruct basically any neuron we want. It just takes time and effort. And so we can identify and reconstruct single LPC1s, as you can see here. And then a single LPC1, because we've mapped several and reconstructed all their inputs, receives plenty of inputs from T4 neurons and T5 neurons, and they receive inputs from the B type, the ones which are selective for back to front motion as expected. A single LPC1 receives inputs from something like 30 T4 and T5 cells. In addition, what we find is we find plenty of inputs from other columnar neurons. In this case, a type of cell called a TMY cell. And these neurons bypass the motion detection step and get direct inputs from the monopolar cell, so roughly the fly bipolar cell. So these neurons receive faster inputs. They are accordingly tuned to faster motion, to faster speeds than the T4 neurons are. And so we have this direct sort of parallel pathway for conducting faster visual information into these neurons. And so I think it's the discovery of these parallel pathways which may help us close this gap between the behavioral and the neuronal speed tuning. And so just to really cut to the chase and summarize, because I threw a lot of things at you quickly, we've discovered this pathway which links translational motion vision to the control of forward walking. And actually the evidence is pretty straightforward. So when flies see backwards motion, they slow down. These LPC1 neurons, they encode backwards translational optic flow. If you silence the neurons, the flies don't slow down when they see backwards optic flow. And when you activate the neurons, the flies do slow down. And so it really looks as if we have a really key piece of this backwards motion mediated slowdown behavior. Now, is this a behavior I would have chosen to study? No, but this is really the beauty of doing biology with, let's say, an embracing ignorance approach of doing a screen, right? That the brain tells us what neurons we should work on rather than the other way around. But back to the speed tuning thing, we have this preliminary evidence for a parallel faster motion pathway. And so that means that this behaviorally measured speed sensitivity is likely enhanced by neurons that are faster than T4, T5. And so we've got some work to do to figure out how they synergistically interact with T4, T5, because that is not actually that straightforward. But of course, in an EM volume, you can keep going. And so we've reconstructed here the primary target of the LPC1 neurons. There are more, but this is just one that we particularly like. And so this sort of implicates a higher brain area, the so-called anterior ventrolateral protocerebrum, as an area for higher visual processing. And so what's quite interesting is what we don't find, right? So for these backwards seeing visual neurons, we are not finding strong connections to any of the neurons of the central complex or a lot of connections to descending neurons. And so what I interpret from this is that in our quest to understand these brain-wide behavioral circuits that mediate these visual responses, we really need to keep mapping, right? And so we actually don't know, we don't actually have many complete circuits outside of that escape behavior I showed you. And so this is just a fun conjecture to think about. What I think this means is that much more of the brain is visual than we had previously appreciated. But once you leave the visual system, you've left retinotopy behind. And so the neurons don't really look visual, and you wouldn't know it without this connectomics approach. And so we have a related paper for a feature detecting pathway that's up on bioRxiv and thinking about how spatial vision is implemented in the absence of retinotopy that you might find interesting. Right, so I'm going to end with just one fun thought. So everything I've talked about today has been in the feed forward sort of traditional way of thinking about the brain, right? The so-called neuronal control of behavior. Let's go from the photoreceptors and work our way inwards. And of course, we know that this isn't quite right, right? And it's more and more important to start thinking about not only the neuronal control of the behavior, but the behavioral control of neurons. And so our favorite version of this is in thinking about the octopamine system. And so here are some octopamine neurons where we're just indicating where their presynapses are. And so these are neurons which receive their inputs in the central brain and project outward, where they contact many, many of the neurons I've told you about today and many others, and differentially modulate their activity, right? And these neurons are driven by behavioral events. So there's a lot of really exciting work on this. But this is at least the other piece of the coin, or it's a 8-sided die or whatever it is, right? That at least in part, this mismatch between the speed tuning in behaving flies and in the neurons is explained by these changes in behavioral state, which is again in part accounted for by this octopaminergic modulation. And John Todd Hill had a beautiful paper on that, but there's a lot of really excellent work on this. Okay, and so with that, I'm going to skip the very last 2 slides and just go to my acknowledgement slide. Oh, sorry, I have one more thing, I apologize. I just wanted to say this, that I get a few emails every week where people are asking me for parts, and I'm very happy to give them out. It's just taken a while to organize this, but Frank Loesch in the lab has been organizing a GitHub where we've put most of the main components that people tend to ask us for. So things like our fly holders, the fly on the ball setup, et cetera. And in the next year, we'll have a couple methods papers out. So we're trying to make these methods easier to reproduce and cheaper and more accessible to people. And we'll have an update on our visual displays. So keep an eye out for those methods papers. And then very quickly acknowledge all of the really amazing and talented people that I've been fortunate to work with. And just briefly highlight Jim and Shunze, who did the bulk of the T4 circuit mapping work with amazing lines from Alyosha and a lot of support from Ed. Eyal did this heroic work on the whole cell patch clamp recordings. Kit is an amazing person just to have around. And then Matt and Jessica were the people behind the LPC1 work. And of course, amazing support from colleagues at Janelia and HHMI. Thank you so much for your attention, and I'd be happy to take any questions.
All right, thank you. Let me start going through the questions. I have a couple questions, but I will save those to the end. Not take advantage of my position too much. So the first is from John Hernandez, who says, fantastic talk, of course, great data. It says in the mammalian cortex, especially the prefrontal cortex, there are neurons that display directional selectivity. These neurons don't seem to contribute to the decision-making process, given that preferred directional preference of these neurons doesn't really correspond with an optimal outcome or reward. Do you think that directional selectivity you describe in T4 and T5 is important for reward seeking at all?
So that is a good question. I mean, so I don't have anything terribly clever to say about this. I would just think that something like reward seeking would involve brain-wide coordinated activity. So it wouldn't be surprising if in the context of a reward-seeking behavior where the animal is being conditioned to prefer a single direction of motion, you would see asymmetries which are not there in a naive animal. But those are certainly not experiments we've done. And in the fly brain, I think the place to look for that would be further downstream. Even though I've said there are these neuromodulatory signals going all the way back to the visual system. So I think one of the opportunities and challenges we have in the fly is that we can record from any part of the fly brain. And so just because we see things in the visual system, right, we're seeing more and more evidence that neurons in the visual system know exactly what the animal is doing, and that they're as good at encoding these behavioral events as they are at encoding their so-called preferred visual stimulus. And so I think we do need to move back and forth in vivo behaving experiments between our early parts of the nervous system and some deeper neurons, once we knew all the right reward neurons to be looking at. But there's dopamine in the visual system. There's all kinds of feedback. So
has the dopamine in the visual system been studied at all? I mean, is it like dopamine in the visual system like the mammalian retina, where it's like a gain control mechanism, or is it?
We don't know.
Okay.
We don't know. There's one of each, right? There's some serotonin neurons, there's some dopamine neurons. So, I mean, the homologies are interesting, right? But someone actually needs to work this out.
Okay, so the next question is from Pavan Kumar Kaushik, which asks, again, lovely work. Real-world scenery has diverse spectral strengths. So unlike a clean square transition with a sharp edge, the input will be, he describes as quite wiggly due to the numerous edges and varying contrasts. So how do these neurons cope with such naturalistic input where it's not quite as obvious?
Right, so of course, no, that's a great and super interesting question. I mean, I would say we consciously, right, we're aware and inspired by naturalistic stimuli, but we consciously used this reduced approach, right, so that we can actually make strong claims about what the neurons are doing, right, by being able to decompose the stimuli into its parts and then put it back together, which you can't really do as well with naturalistic stimuli. I will just say, even within natural scenes, right, I think visual systems and behavior is really primarily dominated, right? So natural scenes have this power law distribution. And so it's the low frequency stuff, right? Which is the large blocky things, which are dark and bright, which tend to dominate these behaviors anyways. So again, I don't have so AL did not do these experiments with broadband stimuli. So I don't have anything terribly clever to say about that, but I would be shocked if, without just elaborating the filtering that goes on upstream, and there's really been beautiful work from Axel Bort's lab and from Damon Clark. So there's a lot to be said about reconciling these two ways of thinking about things. I don't think there's like a big hole in the way we have to think about how the neurons work, but there would be some careful work to actually graft the two ways of thinking about how the T4, T5 neurons work together into a unified understanding.
So there's another question on the T4, T5 cells from Dinesh Nadasan. Oh, just updated. I lost it for a second. There we go. Asking about the linear summation of the spatial filters. So the spatial filters require this specific dendritic synapses of input onto these cells. Do you have any idea how this synaptic specificity is formed to ensure this reliable motion from fly to fly?
Sorry, this is a question about development?
Yeah.
Yeah, so I mean, maybe it's a question about development. So I don't think we have very good evidence for strong activity-dependent contributions to this patterning. I mean, of course, that experiment hasn't been done. We just know that flies who've never been exposed, who've been dark reared, right, essentially behave the same way, right? So there might be very small differences, but we've missed them if they're there. And of course, no one has done a connectome on a fly that never saw a bright edge in its life. I would just say that the patterning seems to be extraordinary, and there's a large number of labs who are quite interested in that developmental program that generates, especially those incredibly specific connections, like at the very tips of the dendrites, right? So the thing that makes it remarkable is that those neurons are everywhere. So those columnar neurons, right, are really shooting through those dendrites at every location, at every column, but they're only forming synapses at very specific locations, right? So I think that's a beautiful opportunity to understand that developmental mechanism. And it works. I didn't show this, but for neurons with different orientations of the dendrite, we find the exact same pattern. And so this has been beautifully described in the EM work from Janelia.
And kind of a related question, Georg from Munich asks, do you have any idea or hypothesis what the cells that synapse onto the tips of the T4, T5 cells could be doing? I said MA9 and TM9, since this part of the dendrite seems to be indispensable for generating direction selectivity.
Yeah, yes. No, so we tried really hard to understand it. I'm guessing I know who the question is coming from. We have in that 2017 paper, we have phenotypes, right? We actually did quite a lot of digging to investigate the role of MI9, the input on the tip. It's a weird one. It's an off cell. It's inhibitory. We used a whole range of visual stimuli. We actually did some RNAi to remove it. It's a glutamatergic neuron, and we removed the glutamate gated chloride channel, and we found that it phenocopies a specific behavioral effect. So we did a lot of detective work about that. I still don't really know what it's all good for. I have a hypothesis that it might be used to sort of sharpen up edges and might help actually keep things faster. But it's not it's far from solved. And AL doesn't find really striking responses to visual stimuli all the way at the tips to typical on and off stimuli. So it's a mystery. That's what I meant, generally speaking. Like, you can do this detective work where you try to do every one of these manipulations and then just use, an extreme logical exercise to reason about all the results and try to explain everything. And I feel like you can get 80% of the results to sort out in a logically consistent framework. And then there are a few things which kind of poke you in the eye. And that's where we are for that question.
So let's see. This person doesn't have a name showing up, but they ask if there's an integrated network that works in parallel to these neurons to respond to color vision, either separately or integrated with the moving looming stimulus.
Yeah. So definitely. So Kit Longden in the lab has been working on the interaction between the color pathways and the motion pathways. And so we can say that, let's say the motion pathways are enhanced by color vision. So, but what we mean by color is circuits which are downstream of photoreceptors, which are have different bandpass characteristics, right? So they're tuned for, let's say, UV light in the case of the short wavelength ones, and some blue and even reddish light for the longer wavelength ones, right? So those signals do get into the motion pathway. So T4 and T5 are privy to some amount of color information. It would be kind of a terrible not to take advantage of that because it's already there. And then, but most of those color signals do seem to go towards the lobula to these feature detecting pathways where they do interact with things like looming detection. And so that is a brave frontier for people who want to work on it. I think there's a few labs who are taking up that color pathway work.
Next question is from Matt Summers, who asks whether the directional selectivity of T4 and T5 cells varies over the range of temporal frequencies for which they are tuned.
Does the directional selectivity, I mean, it's pretty constant until it falls off dramatically at faster speeds, right? So yeah, that's about all I will say. What we've never done is we've never done the experiment with EFIS. So we've done it in calcium imaging, in animals that are behaving, or in the case of octopamine modulation, right? So the temporal selectivity, the temporal frequency is pushed. Here, I can just, I'm not showing my slide, but I have a slide on it. It's not important. There's a 2018 PNAS paper about behavioral state modulation, and we show that you can push the temporal responses of T4 quite a bit, either when the animals are behaving or with octopaminergic modulation. And so that's a little too simple to explain this gap. I think it explains about half of the gap between the neurons' temporal frequency tuning and the behavioral one.
Then one last question about T4, T5 cells, and a couple other questions, and then we'll finish up. So last question about T4, T5 cells comes from Shubham Rathore, I hope I'm pronouncing that correctly, who asks whether T4 and T5 cells are known to directly regulate each other.
Do they directly regulate each other? So I don't believe so. I mean, what happens, which is quite interesting, is, I mean, so what I'm thinking about is whether there are not well-described feedback mechanisms in the lobular plate, right? So their dendrites are actually totally different neurophils, right? So, I mean, of course, there's a delta notch interaction during development. So that's a different thing. But in the adult, when they're all patterned and set up, The question is, do they modulate each other? So not directly. What they do is they work, let's say, cooperatively through inhibitory interneurons to generate what you could think of as a directional or motion opponency onto the postsynaptic neurons, right? And there are a couple of weird things which have not really been well described yet that are happening in the axon terminals of these neurons that we're aware of through connectomic studies, but the functional work hasn't caught up.
So Umesh wants to know if you've come across any small field sensitive neurons which don't get local inhibition.
Any small field? No, I pretty much think all neurons get inhibition.
Okay.
Right. Yeah, that seems to be a general rule. I mean, in fact, most of these neurons are pretty nonlinear. Once you get away from the second and third, order cells in the visual system, right? It's actually pretty hard to get them to respond to anything, right? And so that is some evidence that they're powerfully inhibited all the time by just about any visual stimulus, right? It takes a while to kind of lock, find the key to their little stimulus selectivity lock.
Last questions comes from Brian Jones, who asks your thoughts on an integrated modeling environment for processing the current flow, and it's the logic for the complete circuits.
I think it's a good idea. I mean, we all have to decide when we do modeling, right, what the point of the modeling is and whether, I mean, I tend to be a little conservative in thinking about this, which is, I think we don't yet know enough, but that's always going to be my inclination. And so we've tried to do our modeling at a very small scale, right? So we are very explicit, right? When you do a model, you should think about what the goal of the model is. And so the goal of our model was really to ask whether, can you use the static receptive field mapping of the neuron to explain its responses to dynamic stimuli? So that was really full stop. That was the goal of the model. And we resisted the let's say, building a giant model of the entire motion pathway, right? Because our model already had 16 parameters. That was kind of enough for me. It would be hard to think about how, in a reduced way, to not overfit, all of these data. But we've done different models for different purposes. Other people in the field have. I think we're pretty close. I mean, if someone wanted to work through more properties of the visual system, right? In particular, we currently don't appreciate fully the impact of wide field processing and feedback mechanisms, lateral interactions, which are a source of a lot of inhibition. And so that's the one piece of it which we don't appreciate well enough. But the small field local stuff, I think there's certainly enough to work with if the goal is to predict T4, T5 responses. And people have approached that in different ways. So the Borst lab, Damon Clark, some of our modeling work, we've kind of done it in a reduced way. But yeah, I think if a computational lab wanted to build a full model, we'd be happy to feed them, insights.
Yeah. I actually have one last question, which is in your model at the beginning where you have basically a better dynamic range or a better difference between the motion processing, the direction processing in the contralateral and primary directions. What is it about the model that gives you that better, I guess, contrast is the word, between the directions? And do you think that's the best you can do, or do you think there's something better in like a simple circuit?
Sorry, I just want to make sure I got it. You mean I would say, so let me just say something and tell me if this sort of answers your question. One of the things I didn't appreciate when we started working on this stuff, right, is you kind of have to think fully about what a neuron responds to as well as what it doesn't respond to, right? So I think people who are sort of searching for best stimuli, a lot of this happened, if you're recording single units in a vertebrate, a bird, a monkey, whatever, right? You spend all this time hunting for stimuli which will drive your neurons maximally, elicit the highest firing rate, right? But what I think is less appreciated is it's more interesting for the question of selectivity to think about the sort of the null space, right? Like, what is this whole set of stimuli that are pretty close to the stimulus you think you're studying, but that the neuron ignores, right? And so directional selectivity gives you a really sort of crisp statement of that issue, because it's really just if we agree what the goal of the computation is, right, which is a big step, right? But if it's really just to say right from left, right, then it's very clear how well and how selective a neuron is. Once you start to get into feature detection, right? I think the number of conjectures and you have to make are larger. So I suppose one could imagine designing a nearly perfect directionally selective element, but it would have some shortcomings in other ways, right? It would be very sensitive to noise, right? So, I mean, one of the beautiful things that's going on in the fly visual system, which I would love to sort of write up in a review at some point, is that like all the basics, anyone who's ever learned any signal processing, right? A little bit of filtering, all of that, right? Like all of that is implemented in the fly visual system, right? So if you think of directional selectivity as computing a derivative in space and time, as everyone who's taken a derivative of sample data knows, it's noisy and you amplify noise in that process. And so the way to deal with that, right, is of course you filter your data, smooth it, downsample, and then compute the derivative, right? And that's exactly what the fly visual system is doing. So I think, you know, there's some noise robustness which limits just how accurate you would ever want a motion detector to be, right? Because it would have this real confound of being driven by noise, or like, you know, if you're staring up at the sky where you don't have strong contrast, you don't want your motion detector to go crazy, right? So you have to sort of limit the performance for those reasons.
Okay, fair enough. Okay, thank you again for speaking. For everyone, next week, Deborah Gordon is speaking on ants and ant neuroscience. So I hope you come and hear about that. And thank you again, Michael.
Okay, thank you so much. Take care, everyone.